We're here to fill you in on two of the most important plays of the cell cycle, mitosis and meiosis.
Mitosis produces the trillions of somatic cells making up our bodies, and it also replaces dead and damaged cells.
Meiosis produces gametes, eggs or sperm, occurring in the gonads. Genetic recombination is also an important aspect of meiosis, resulting in variation of offspring. Natural selection throughout evolution causes the offspring with the most favorable traits received through genetic recombination to survive.
For future reference, a drawn X is a chromosome, and a double X is a double chromosome.
We'll start off with mitosis. 2n46, 2n46, hike.
The cell cycle is composed of interphase, mitosis, and cytokinesis.
Interphase has three parts, G1, S, and G2. These parts are like a warm-up to mitosis for our team, the cell. In G1, the cell grows. In S, it continues to grow as it copies its chromosomes. In G2, it grows more as it completes preparations for cell division.
After warm-up, our team is ready to go. The team has now doubled its number of players, better known as its chromosomes.
The first stage of mitosis is prophase. The chromatin fibers become tightly coiled and condense into chromosomes, and the nucleoli disappear. The nuclear membrane breaks down. The centrioles move to opposite ends of the cell. The duplicated chromosomes are identical sister chromatids joined at their centromeres. The mitotic spindle, shown in pink, begins to form.
After this part of the play is completed, the team moves to the next stage, metaphase. This is the longest stage of mitosis. The players line up on the metaphase plate, an imaginary plane in the middle of the cell. The spindle also continues to form. The double-stranded chromosomes are attached at the centromeres to the spindle microtubule.
The team then moves into the shortest stage of mitosis, anaphase. It begins when the centromeres of each chromosome separate. There are now twice as many chromosomes as there were in metaphase because each sister chromatid has its own centromere and is now a chromosome. The two daughter chromosomes begin moving towards opposite ends of the cell. By the end of anaphase, each end of the cell has equal and complete collections of chromosomes.
The team then moves into the final phase of mitosis, which is telophase. Two daughter nuclei form in the cell. The nuclear envelopes form, nucleoli reappear, and the chromosomes are less condensed. After mitosis is complete, the team is not finished yet. It still must go through cytokinesis, which is usually well underway by late telophase. In animal cells, a cleavage furrow forms and pinches the cell in two. In plants, a cell plate forms.
Cytokinesis is comparable to the victory lap or the shaking of hands.
The final result of this play is two identical teams with diploid number of players.
Now you know all about our first crucial play to success. We will now move to our second play, meiosis. N23, N23, hike.
Meiosis is a little more complicated and is divided into 2 rounds of chromosome separation and 2 divisions, but only 1 replication.
Meiosis 1 involves separating homologous chromosomes. It is preceded by an interphase similar to that of mitosis interphase.
In the first part of this play, prophase 1, chromosomes begin to condense and crossing over is completed. This is the exchange of corresponding alleles by non-sister chromatids. Centrosome movement, spindle formation, and nuclear envelope breakdown occur as in mitosis.
The next part of the play is metaphase 1. Pairs of homologous chromosomes are arranged on the metaphase plate. One homolog is attached to the microtubules from one pole, while the other homolog is attached to the other pole.
On to anaphase 1. Proteins responsible for sister chromatid cohesion break down, allowing homologs to separate. Homologs move towards opposite poles. Sister chromatid cohesion persists, allowing them to move as one unit.
Next is telophase 1. At the beginning of telophase 1, each half of the cell has a haploid number of replicated chromosomes, each composed of 2 sister chromatids. Cytokinesis usually occurs simultaneously with telophase 1. In animal cells, a cleavage furrow forms, and in plant cells, a cell plate forms.
No replication occurs between meiosis 1 and meiosis 2. The cell then goes into meiosis 2, in which the team separates sister chromatids. As a side note, all of the stages that occur in meiosis 2 happen in the 2 cells that resulted from meiosis 1, as in all of meiosis 2 happens twice simultaneously. For spatial purposes, we only illustrated each of the stages happening once.
In prophase 2, a spindle apparatus forms.
In metaphase 2, the chromosomes are positioned on the metaphase plate. The sister chromatids are not genetically identical because of the crossing over in meiosis 1. The connecticers of sister chromatids are attached to microtubules extending from opposite poles.
The team then moves to complete anaphase 2. The chromatids separate after the breakdown of proteins and move toward opposite poles as individual chromosomes.
In telophase 2, nuclei form, the chromosomes begin decondensing, and cytokinesis occurs.
Result of meiosis is 4 daughter cells, each with a haploid set of unreplicated players. Each of the 4 daughter cells is distinct from the parent cell and each of the other 3 daughter cells.
To sum up our plays, we will recap some similarities and differences between them. DNA replication occurs in both plays during interphase. Mitosis has 1 cell division, while meiosis has 2. Crossing over occurs only in meiosis during prophase 1.
Mitosis produces two diploid cells identical to the parent cell and to each other. Meiosis produces four haploid cells containing half as many chromosomes as the parent cell, which are not identical to parent or other daughter cells.
Both of the plays that we covered today are extremely important. Mitosis allows our bodies to repair dead or damaged tissues, as well as allowing us to grow from a zygote to a multicellular adult. Without meiosis, reproduction would be impossible and mitosis would be rendered irrelevant. Therefore, knowing the details of both of these important cycles is essential to understanding the biology of life.